Welcome to the lecture. In this lecture, we are going to discuss the concept that is transition metal complexes as a chemical nucleases. Now, in this lecture, we are also going to discuss what is meant by the nucleases, their types, and their subcategories. So, let's discuss. Transition metal complexes as a chemical nucleases. As we know that the nucleases are nothing but the enzyme which can rupture the DNA bond. So if suppose there is a DNA and RNA bond, as we know that this is a 3' end and that is a 5' end. Again, this is a 5' and this is a 3' end. In this strand, there will be this, in this portion, there is a base pairs are available. That is adenine, guanine, cytosine, thymine and so on. Now, nucleases is an enzyme which can rupture the DNA bond either by 3' end or by 5' end or in the middle portion. So, for that purposes, if we rupture that bond, that is and if we use the chemical nucleases in this portion, so if you want to rupture that type of bond, then the structure may be as you see, that is this portion is totally blank. So here the bond here getting breaked and we have appear, that is breakage of this DNA bond or the cleavage of DNA strands here. So likewise here we can say that nucleases is enzyme which cleave DNA strand or bond by cleaving phosphodiester bond between nucleosides of nucleic acid. So here, if you see, this is a DNA strand and it can easily cleave by using this nuclease as an enzyme, and it can cleave the bond as or the rupture the DNA strand as like. Now, nuclease is an enzyme. As we know that it can cleave the DNA bond and it generally cleaving the phosphodiester bond between nucleosides of nucleic acid. Now, depending upon the mode of action or the target centers, it will be divided into two types, that is endonucleases and exonucleases. Endonucleases means can target or can break or rupture the nucleic acid from the enzyme. So that's why it is called as a from the middle side. It is from the middle side, whereas exonucleases can digest the nucleic acid from the inside. So this is a endo and exo. We know that the exo is in outer or an outside portion, whereas the exo is in outside portion, whereas the endo is in inside portion or from the middle portion of this base pairs. If suppose the rupture or the nucleases can act here, from then there is a exonucleosides and nucleotide or the nucleases that rupture that nucleic acid, means that RNA and DNA from here, then this is called as an endonucleases. So this is the difference between the endo and exonucleases. It is further subcategorized into DNA or RNA, and mainly it is depend upon deoxyribonucleotide and oxyribonucleotide. So now let's discuss in detail the reactions of metal complexes, or the reaction or the interaction of metal complexes with DNA. Let's discuss the reactions of metal complexes with DNA. As we discussed earlier, that is nucleases is enzyme which can cleave or rupture the bond or the strand or the fragment of the DNA, that is either exo, or means that is inside, or in middle portion, that is an endonucleases. So what is meant by the chemical nucleases? Actually chemical enzyme nucleases is in chemical nucleases are the some synthetic enzymes which can cleave the DNA bond. So that's why this is called as in chemical nucleases. Now, as we know that there are various diseases which can very much affect on DNA and RNA, that is cancer. We know that the replication of DNA causes the number of uncontrolled growth of that cancer cells or the tumor cells. If we arrest that growth, so that's why we have to stop the growth, or if we stop the replication of DNA in designed manner, then it is very easy to stop the uncontrolled growth of that tumor or the neoplastic cell. How the reactions or the interactions can affect the DNA? Let's see that the DNA is a fruitful target for DNA for metal complexes. DNA can function as a ligand, either through either through reaction with phosphate group or coordination with base with bases, whereas non-covalent lead to additional targets and greater specificity through an interaction in DNA group and base track. DNA group and base track. So generally, if suppose we use the chemical nucleases, it will react with DNA. It is either, or interaction of the metal complexes is by the covalent or non-covalent. Here this can attack or this can interact with DNA in that manner. Either it will be in from group, this deepness of this two strand, or the deepness of making the position of this group, and this is the base track. The portion, or this is a narrow portion in between these two strands, or this narrow portion is developed by these two strands, is known as a group. So here this metal complexes are interact with this DNA, that is either by group or by the base track. Now first reaction, or the reaction of metal complexes with DNA, that is covalent interaction. That is a covalent interaction. Generally in covalent interaction, what happened? It has been widely accepted since the DNA is the biological target cisplatin. As we know that the cisplatin is an anti-cancerous agent, it can bind to the DNA in that manner. That is, platinum is in center. Here 2 ammonia and 2 chlorine atoms can bind this cisplatin in these 2 strands of DNA. Generally, cisplatin is more effective than transplatin because the positions in cisplatins are the cis means that these ligands are in the same positions and it can perfectly bind the DNA strands, whereas the transplatin cannot, due to transposition of this respective ligand, it can not properly bind to this respective nitrogen bases with DNA. So that's why the cisplatin is a more effective anti-cancer agent, and this is nothing but the metal complex also. Cisplatin is a metal complexes, and that's why it can target biological target of this cisplatin. Now let's discuss this interaction. Can propose interest and cross link between the N7 atoms of, that is nitrogen bases, adjacent, N7 atoms of adjacent guanines and cisplatin base and cisplatin based. The study of, on the result of numerous biological studies. So these are the reaction of metal complexes with DNA, that is a covalent interaction or the covalent reactions. Now let's discuss the next one, that is intercalation. Intercalation, metal intercalation is DNA binding mode, DNA binding mode. So that has been extensively studied. As with organic intercalators, this complex containing aromatic that can stack with DNA pairs. That can stack with DNA base pairs. Generally, organic heterocyclic ligands are holding in between these strands, or the metal complexes that can hold between these two strands, which can react with this DNA or for further reaction. However, the lack of site specificity inherent in intercalation by planar complexes. So that generally it is found that the planar complexes are more preferably bind to the DNA base pairs or with DNA strands. So these are all about the reactions or the interactions of metal complexes with DNA. In that we have seen the covalent interaction, second that is a intercalation interaction, and the third one that is a insertion of the metal complexes in DNA. Let's see the reaction of metal complexes with DNA, that is a insertion. So in DNA metal complexes, in metal complexes we know that there are various complexes which can interact or inserts in DNA base pairs. Which one are they? Let's discuss. The criteria for insertion in DNA base pair that it can perfectly bind or find the position or the specificity in DNA base pairs. But generally what happens, rhodium complexes or the rhodium metalloinserters are bind to mismatches in DNA binding with respect to high specificity and selectivity. And selectivity. The binding of complexes with DNA base pairs is depend upon the size and their arrangements in these base pairs. Generally, rhodium complexes are not too much there is a mismatch for binding with this DNA base pair, whereas the platinum complexes, as we know that the cisplatin is an anti-cancer agent which can perfectly bind the base pairs, and it can effectively stop or control the replication of this DNA for the uncontrolled growth of the new plastic cells. Now, the cisplatins or the platin complexes have considerable DNA binding property, as it with respect to high specificity and selectivity. These are the insertion or the reactions, and these are the inserters that can insert in DNA base pairs to bind in between these two strands. So these are all about the reactions of or the interaction of metal complexes with DNA base pair. Now we will stop here. In next lecture we are going to discuss the nucleus of activity of the copper phenanthrone complexes.